Magnetotactic organisms. I'm going to hand over to you because I love the subject so much. I'd rather just hear you talk about it. But I can't believe that there are microbes with tiny magnets inside them. That just sounds amazing. But do you have a presentation? Should we just check if we can share the screen? And I think I've given permissions, yes. So it should be okay. Right, fantastic. I'm going to turn my camera off, but I'll still be here. Let me know when things are over and I will have probably thousands of questions for you, but yes.
Okay, I hope so.
Begin.
Okay, so thanks for having me here, Joe. It's really nice for the invite to talk about a topic that I'm really passionate about, and I have developed some research during my time as an undergrad, in my master, and also during my PhD. And this topic is magnetotactic organisms. And I really like to start this topic talking a little bit about how this was found out at first, because I think it's a really tricky story. So the first paper that came out was in '75, and it was published by Richard Blakemore. He was a scientist in Woods Hole Oceanography Institute in Massachusetts, in United States. And his description was really simple. He observed some tiny microorganisms that once he observed in the microscope, if he puts a magnet on the side of the drop, he could see that the cells were migrating towards the magnet. And once he flipped the magnet, the cells were going to another direction. So that was the first report. It was a big thing, as you can imagine. But later on, Richard Frankel, one of the first person who actually developed more studies in magnetotactic bacteria, found out that in 1963, there was not a paper, but a scientific report that was describing some behavior really similar to what Richard Blakemore had found out later. So this finds are here. It was made by Salvatore Bellini, a medical doctor in a university in Pavia in Italy. And what he described was bacteria that were sensitive to magnetic fields. And he described this as a magnetosensitivity or sensibility. So he observed exactly the same thing. And what really intrigues me is that if we see the way he described this behavior in this picture on the left, it's really similar with the models that we develop and we observe nowadays, or when we start to have more tools to investigate a little bit more, as we can see here in this model on the right, that I will talk a little bit more about in a couple of minutes. So now starting with the topic itself, what are magnetotactic bacteria, or MTB for short, as we call them? At first, this is not a taxonomic classification. So magnetotactic bacteria are really diverse. They are spread to the tree of life, as I'm going to show you guys. But the common characteristics that we have is that all of them are gram-negative. So they have cell wall structure as gram-negative bacteria. They are found in aquatic environments. So we can find them on the sea or on fresh water, basically virtually in any aquatic habitat. And all the bacteria that we found so far, they have a metabolism that is sensitive to oxygen. So they are anaerobic or they are microaerophilic. They need really tiny concentrations of oxygen to live. All of them have a motility biflagella, so all of them can swim. And of course, the signature of the group are these organelles they can produce. There are the magnetosomes. There are the tiny crystals that in these pictures in the TEM, we can see the cell here, the cell shape, and these black structures inside the cell are the magnetosomes. Each individual crystal of this is called one magnetosome. Here we can see some of the diversity. Then we're going to talk a little more later as well. As I was saying, this magnetotactic bacteria is not a phylogenetic classification. Here we have a beautiful tree of life where we can see that is highlighted now where we already found magnetotatic bacteria. Most of them are in this region of the tree. They are belonging to the Proteobacteria phylum. And more precisely, most of them are in the alpha Proteobacteria and the delta Proteobacteria, but also we can have some here in the PVC superphylum. And later we have some clues of a magnetotic eukaryote that we'll be talking at the end of this presentation. So they have a phylogenetic diversity and also a metabolic diversity, because we can find these organisms in different types of environments. So in environments with a high concentration of salt, we can find them. Also in ponds and lakes with a high pH, we can find this group of bacteria. Also in lakes and also in the sea with really low temperatures, we can find them. And also in the places with high temperature, like the Death Valley in the United States. Talking a little bit more about the magnetosomes itself, they also present some diversity. So these crystals that they can produce, they are composed of two different parts. The mineral part, there's the crystal, the magnetic crystal itself, that can be of greigite, iron sulfate, or magnetite, iron oxide. And one crystal only can have one type of mineral. And usually the same bacteria produce only one type of crystal, but we have some species that can produce both crystals depending on the conditions they're growing in. And all these crystals have an individual membrane that surround the crystal. So here in the image, you can see different shapes of crystals, right? On the left, we can see the cubooctahedral crystals. In the middle, we have the prismatic crystals. They are more elongated, as you can see. And on the right, you can see the anisotropic crystals. These ones have a bullet shape. And surround each one of these crystals individually, you have a phospholipid membrane with a lot of proteins inserted on it. The crystals can also be organized in different ways in each one of the cells. Here on the left, we can see a cocci with two chains of magnetosomes, as we can see, and they're pretty set apart. The cell in the middle have one single chain close to the middle of the cell. I would say that this is the most common organization that we see, at least in the papers that were published. And on the right, we can see multiple chains of magnetosomes inside one cell, but these chains are clustered in the center of the cell. I'm talking a lot about these chains of magnetosomes. As you can see, they're pretty much organized like this, but we also have some cells that organize the crystals, apparently not in chains, but in clusters, like here, and this one on the left, as we can see. This is a multicellular form that I will talk a little bit more later, but they also, at a first sight, they have some disorganized magnetosomes inside the cell, but they are not disorganized, I promise you, I will show you later. And on the right, we can see this cocci. They have kind of some structures in chains along the whole surface of the cell. One common question that people ask is, how the cells can produce the so perfect structure. So these crystals, they're not only crystals, but they're also magnetic. So the magnetosome itself has the phospholipid membrane around it. And in this membrane, we have specific proteins that allow this biologically controlled mineralization of the magnetic crystal. This is also pretty diverse when we talk about magnetotatic bacteria, because what I want to show in this picture is that different classes of magnetotatic bacteria have a different arrangement of genes. But of course, we always have some conserved genes that are responsible for the main structure of this membrane and also the crystal. So in purple here, we have the conserved MEM genes, the genes associated with the magnetosome production. We have some other MEM genes that are not conserved in all bacteria, but they are present in almost all of them. We have some MAD genes. These genes were first described in the delta Proteobacteria, as we can see here, but now we also find them in the Nitrospira phylum. And we have some MEM genes, the genes that Until a couple of years ago, we're only finding nitrospira, but now we already find some MAM genes outside this phylum, magnetotatic bacteria outside this phylum. So we can see here that the production of these magnetosomes are pretty complex. And so far, we don't understand exactly how this process works, but we have some clues. And the model that we developed, it's kind of like this. So at first, the cytoplasmatic membrane suffers an invagination with already some proteins on its surface that allows this type of invagination. The second part is once the vesicles form, there is the recruitment of some specific proteins that will start the process of biomineralization. This process starts with the nucleation of a crystal, so we can start to see a really tiny iron accumulation in one of the sides of the vesicle. And then with the help of other proteins, we start the maturation of the crystal. So the crystals start growing. I want to highlight here something really nice that was found out that at this part of the process, we're not sure if in the maturation this already exists, but definitely when the nucleation this already exists, but definitely in the maturation, we saw some proteins that are interacting directly with the crystal to help their shape being controlled and also the size of the crystal in the end. So as you can see here, we have other proteins in this part of the process helping the crystal to stay in shape and in the right size. Also, I'm going to highlight these blue filaments here. They are a cytoskeleton filament majorly composed of a protein in magnetotatic bacteria called MMP. It's an homologue of actin. And this is one of the cytoskeleton filaments that allows those magnetosomes to be aligned in chains inside the cells. And some of the magnetotatic bacteria have proteins that work as a hook, so they can put the vesicle in contact with this cytoskeleton filament. And in the end, what we're going to have is the magnetosome chain in most of the magnetotatic bacteria with mature crystals and all other crystals in other parts of this process of formation. Here is a nice TEM image of a vesicle being formed. So the outer membrane of a gram-negative bacteria is the inner membrane. We can see the vesicle form here. Then we start the nucleation of the crystal. A really tiny crystal is formed. This crystal starts the maturation process until they are full developed here. And we have the full magnetosome with the crystal and the membrane around it. Now, maybe the question that I heard the most is, so it's really nice, these crystals, but why the bacteria producing this? Why they want these magnetic crystals in the first place? So this can be a little tricky, but I'm going to try to explain as simple as possible. In this cartoon, we have the Earth, and it's just like we slice the Earth on half. And what we can see here is that this red arrow is this geomagnetic field, the magnetic field that we are all immersed in this planet. And this little guy here is a magnetotatic bacteria with his magnetosomes on the inside. And this one is a regular non-magnetotatic bacteria. As I was saying in the beginning, we all find them in aquatic environment. And in this type of environment, the gradient of nutrients, or especially the redox gradient, is distributed along the water column, right? So for this guy, if he want to migrate to a more oxidative environment, to a more reduced environment, this is an advantage for him because the crystals make him being aligned with the geomagnetic field, which is almost perpendicular to this gradient on the water column. So this guy, without the magnetosomes, have a three-dimension problem when he want to use his chemotaxis to find a place where it's more oxidative, like here, or more reduced, close to the bottom. And this guy is already on a track, right? He knows, he kind of sense, not knows, but he sense where is up and where is down. So he can only go in these two directions. So he always go up when he need more a more oxidative environment, and he goes down when he needs a more reduced environment. So there's more time efficient to look for these two types of environment that he might need in different times of his life cycle. Here is the oxy-anoxic transition zone. This is the area where we find most of the magnetotatic bacteria. And this makes sense because in this area of transition would be the area to be that will have less effort for the cell to change for a more oxidative or a more reduced environment. Just to give one step further of this, here we have a magnetotatic bacteria. This arrow is the magnetosome chain. It's aligned with the geomagnetic field here. And this bacteria is on the surface and close to the surface, as we can see here. So if you need, in the blue cytoplasm, it's showing that it is in an oxidative metabolism. So maybe he needs to migrate to a region there's more reduced for them. So he will migrate downwards to reach this oxy-anoxic transition zone. Here's the opposite. In red, he's had a reduced metabolism. So maybe he needs to migrate to a more oxidative environment. So he swim in the other direction, away from the bottom towards the surface. This is all really good, but it's always nicer if we can see how they do this. So here we have a drop of a sample from the environment. We put a magnet on the edge of the drop, and the bacteria swim to this edge. When we flip the magnet, we can see the bacteria going to the other direction. If we flip the magnet back, we can see the bacteria swimming again towards the edge of the drop. This is way more simple and nicer to explain magnetotatic organism than the cartoon that I showed before. But this is the behavior that we study and we are looking for these organisms. And also this video is pretty nice because we can see this diversity. We have tiny cells, we have large cocci, some small rods as well. So everything that I explained so far was why magnetosomes are useful for the cells, but they are also really useful for us because we have lots of biotechnological applications where we can use them. Magnetic nanoparticles are used in medicine and in the biotech industry for a while, but magnetosomes actually have a really good advantage if you compare with the synthetic particles. First of all, if you need a membrane around your crystal, the bacteria already produce this membrane for you. So you don't have to put another step on your production for these particles. Also, the control of the structure that the bacteria have is way better than any control that we have. What I'm saying about control, I want to say the shape and also the purity of the magnetite that you are producing. But some of the uses are, for example, we can actually use the membrane when we make the immobilization of enzymes or the recovery of some cells, or even if we want to deliver a specific type of drug, we can guide these drugs. We can put the drug in the membrane or associated with a protein in this membrane and guide them using a magnetic field. But also there is some applications that only use the physical properties of those crystals, like hyperthermia, where you put a external magnetic field and you switch the orientation of the field to produce heat, or even using this type of crystal for produce contrast in images for medical exams or even some lab tests. I told you so far, I was telling you a lot about how diverse this group of bacteria is. But as we can see here in the tree again, they are spread all over the tree of life. But most of the information that we have so far from this group of bacteria come from two species that actually belong to the same. They're really close related. The species are the Magnetospirillum griffiths-valdensin and the Magnetospirillum magneticum. As you can see here, they are pretty similar. And this is why we only have a lot of information of these two types, these two MTBs. These bacteria are really hard to cultivate in the environment. So most of the studies that we have so far use environmental samples. So it's based on culture independent techniques. So what would be the next step of doing this? So our idea was to explore the true diversity of this group of bacteria and find more magnetotatic organisms as possible so we can gather in the most information as we could. The dream goal would be to find an eukaryotic with magnetosomes or an archaea. And also during this process, produce innovation on culture independent techniques so we can apply to other fields of research as well that need information from some organisms that are hard to cultivate. All this exposure of the true diversity of MTB would be really helpful for us to answer this question of how magnetosomes spread through the tree of life. We have some theories of a monophyletic origin. Also, we have some theories about multiple events of horizontal gene transfer. It could be events of conjugation, some vesicles that can transport, or phages that can transport genes, or some transformation events, or most probably all of this together that made all this mess. During my PhD, I was really lucky. My main goal was these three topics here. So to explore the true diversity. So I have the chance to collect samples of a lot of different places and make some phylogenetic characterization associated with the ultrastructure of the cells. Here's a cell that have a kind of weird magnetotatic response, but this is what we usually do. We see how the magnetosomes are organized. We characterize the if we can see the membrane around the crystal, the composition of the crystal as well. Here is another type of characterization of the ultrastructure, where we can see the composition of the crystal that are inside the cell that came from the environmental sample. This one was a bacteria found in an acidic environment. This one, a pretty nice one as well, where we do some ultrastructure characterization of the composition and also the shape of the crystal itself. This one was the magnetotatic bacteria found in Antarctica. And this is something that I really like to show people because it's one of the things that we're aiming for. I think it's a little to stretch a little bit too much, call this an innovation, because a lot of people were already doing this, but it's the association of some phylogenetic or even genomic information with the ultrastructure of an organism that is not cultivated. So what we did here is to use fish. We designed some probes based on the sequence that we have from the bacteria that we collect. And then we can identify exactly which bacteria belong to that group in our sample. So this is pretty straightforward and people use all the time to see the shape of the bacteria they are studying from the environmental sample. And what we did a little bit further was to associate this phylogenetic characterization in the same sample with some ultrastructure. So I think this could be really useful in the future, especially with all this genomic data that we are developing, this metagenomics that we have a lot of information about the gene content of some organisms that cannot be cultivated, but we don't have any clue of the shape or how the cell is or what type of features the cell have in their cytoplasm. So this association with genomics and the ultrastructure characterization is always something that I think is exciting. As you guys saw, there is a lot of different types of magnetotatic bacteria. So I choose two types of magnetotatic organisms that I want to show you. So I'm going to use these two as an example of this whole group. They are pretty different from average organisms, I would say. The first one is a multicellular magnetotatic prokaryote, MMP for short. And this is exactly what the name is saying. This is a multicellular prokaryote. So it's kind of a multicellular bacteria. We have two types of this MMPs. A spherical one that we can see here on the left. Each individual have approximately between 60 and 100 cells, and we also have an ellipsoidal one, MMP, that we can see on the right here. And this one, the cells they have are larger, so they have less cells per organism. From the spherical MMPs, I would say that this Candidatus magnetoglobus multicellularis are the most characterized, that we have more information about. And what we have here is the magnetosomes of this organism, and we can see they're almost always on the edge of each one of the cells. And if we do some analysis, we will see that exactly this, the magnetosomes are always in the edge, and these crystals are gray guy crystals, or not the magnetite crystals, so it's an iron sulfate. This image is pretty cool because with the contrast, we can actually see the membrane around the crystal here that form the magnetosome. The first main question is, why do you classify this organism as a multicellular organism and not an aggregate of unicellular organisms? So some experiments were done, and what we saw and what I want to show in this picture is that this is the whole MMP, and once we put them in let's say in a condition of stress, the cells die and they release all the cells that are part of the organism. So we could think that maybe the cells can live by themselves, but when we do some other types of experiments, when we dye the cells to see the viability of them, we see that once the cells detach from the whole organism, they die. They're not viable anymore. So they are only found in the environment in this condition, in the condition where they are a multicellular organism. Another thing that makes us classify them as multicellular is that the full cell cycle of the cell is complete as a unit. So to divide this organism, all the cells in it have to divide themselves and they stay as a unit. And once they achieve a specific volume or a number of cells in this case, they twist, as we can see in this image here. And then we have the formation of two new organisms. So it's a full cell cycle as a unit, as a single organism. Later, in 2014, the genome of one of these MMPs was available. And what was really interesting is that most of the features that we find in the genome are really similar to the other bacteria that do not live in this condition as a multicellular organism. So we don't have much clue of a lot of the features that they need to do to be a multicellular organism. Another behavior that they have that's a little tricky is that they keep spinning when we put them on direct light. We don't know what this behavior is achieved by the cell, but it's also something that we believe can be correlated with some of these multicellular features they have. The other one is the ellipsoidal MMP, and as you can see already here, the first thing that is a little different from the other one is the structure they have. They have this little aperture in one of the poles of the cell here. And also in the center of the MMP, we can see a region that don't have any cells, but always have some vesicles that supposedly they could use it to communicate between the different cells in the same organism. Also in this ellipsoidal MMP, the Magnetononas hongshanens, we found two different types of cells. So supposedly we could have a type of cell differentiation in the same prokaryotic organism, which will be something really different, especially if they work as a unit. And here we were able to reconstruct the structure of this MMP, and what we saw was something that we already expected. The red ones are the magnetosomes, And we can see that from one cell to another, the magnetosomes keep in a straight chain. So this is pretty good for them to work as magnetotatic, because as a chain, they can respond more efficiently to the geomagnetic field. The other topic that I'm going to talk really quick for you guys are the magnetotatic eukaryote. This is the main reason why the title of the talk is not magnetotatic bacteria, because early last year, a group from France observed a huge cell, a eukaryotic cell that could behave similarly to the magnetotatic bacteria. So they were investigating how this cell looked like and why they can respond to geomagnetic field. And what they find out is this ectosymbiont bacteria, that this magnetotatic bacteria, they're covering all the surface of this protist and allowing this protist to use the geomagnetic field just like a magnetotatic bacteria would use. So this ectosymbiont was studied, and they belong to the delta bacteria class. And because they produce these magnetosomes that are attached to the surface, they allow this protist to respond to geomagnetic field and have a magnetotatic response. And at the same time, I was finishing my PhD, and I observed also this behavior on some of my samples. So again, a large cell responding to the magnetic field. When I flip the magnet, they respond exactly like the bacteria would respond. So I did the same thing. I would like to look a little bit further what we have in these cells. And the first thing that I found out was some structures in the cytoplasms that was not really usually saw in protists. And under the TEM, I could spot some chains of crystals there I never saw before. So doing some more analysis, I found out that those crystals were magnetite by their crystal structure and also by the presence of iron and oxygen and the absence of sulfur. And when I compared these crystals with the crystals that I could find in the bacteria, in the magnetotatic bacteria in the sample, they never clustered together. There are always different groups. And also the shape of the crystals. So we can see here, in B, this is the crystals of the eukaryote, and here you see are the crystals of bacteria from the same sample. So this tricks me to think that maybe they're not eating any type of bacteria. The only way they could have these crystals is if they are producing these crystals by themselves. So going to literature, again, I could not find any crystal that looked like the crystal we found in the eukaryote. When we plot a graph of the mean width and the mean length of the crystals, they cluster outside again. So this crystal were unique. So most probably this protist was producing these crystals, but definitely it was a magnetotatic protist. Yeah, it was published as the last paper of my thesis as a characterization of a protist that could respond to the magnetotactic response. But unfortunately, we have too little of these cells on our sample, so we were not able to sequence the genome to actually look for some genes that could be associated with the genes found in magnetotatic bacteria. I want to thank the team who was part of this work. Mostly of this work was done in Brazil on my former group, the Cell Biology Magnetotaxis lab. Ulysses Lins was my advisor since I was an undergrad, but now the group leader is Fernanda Abreu. I worked with her for a short time in the end, but during my whole career, she always helped me to develop this research as well. The other group members, Adam Hitchcock was the PI in Canada that works with the characterization of the magnetotatic protists. Dennis Basilinski was always really a great mentor to our group and helped us with a lot of techniques. And with some people from the Russian Academy of Science, I had the pleasure to develop some of those techniques for the characterization of the fish with TEM. Now I'm here at the University of Groningen as part of the MOMIC, the Molecular Microbiology Group, under the supervision of the Dr. Dirk-Jan Scheffers. And here's my contact. If you guys need to or want to ask any questions now or later, feel free to contact me at any time.
Pedro, thank you. That was fantastic. You have blown my mind several times, and it's only 9:37 in the morning. Multicellular bacteria with tiny magnets inside them. That is incredible. I mean, so I have so many questions and we only have 3 minutes, so I'll just try and pick a couple. So you mentioned that, you know, they use the magnetic fields on the Earth's magnetic pole to kind of organize their movement so they can get to places, or at least have some degree of now directionality. Does that mean they behave differently depending which hemisphere you're on?
Yes, they behave differently. And yeah, the bacteria that we can find in the north hemisphere, we call them north-seeking, and the ones that we find in the south hemisphere, we call south-seeking. And the most intriguing thing is that if you go to the equator of Earth and sample in a lake, you will find 50% of the bacteria from the south-seeking type and 50% of the north-seeking type. Yeah, so I'm not sure.
It's always 50-50.
In the equator, yes.
The equator. Oh, okay, yeah, because they're kind of making that choice. Okay. The other thing I was thinking was, you know, making these crystals, is it quite energetically costly for these microbes? They must have to spend some of their resources making them. So yeah, does it cost them a lot of resources? I mean, it must be worth it if they're doing it, but it seems like quite a big deal for a tiny microbe to make these crystals.
Yeah, this is an interesting question because we have some mutants in the lab that do not produce the crystals, but we don't see they growing faster or in a larger volume in the same time when we compare with the wild type. And also there is another information is that I show you some cells with a lot of crystals, more than 10, and some groups already find out that they don't need that amount of crystals to respond to the magnetic field. They could use at least half of those crystals. So apparently they're not that they do not cost that much of energy because they're producing more than they need.
Okay. And one final question, if we could just squeeze it in, is you mentioned that there's a few different branches on the phylogenetic tree with magnetic microorganism. So does this mean it's evolved convergently several times, or is there one common ancestor at any point in time?
Yeah, that's the golden question that we want to find out. There is some groups that believe we have a monophyletic origin, where we have an ancestor that was the first magnetotatic bacteria. And other groups think there is a lot of horizontal gene transfer events that spread these genes all over. And that's the main reason why it would be really cool to have the genes of that magnetotatic eukaryote.
Okay, yeah, because if that is a true magnetotactic organism, that would indicate radically different evolutionary pathways to this sort of similar behavior. We've reached the end of our time slot, Pedro. I could ask you questions about these organisms for hours, but hopefully people can ask you questions on Twitter. And if you have any more, yeah, reach out to Pedro. I'm sure he'll be willing to chat about that in endless detail in Twitter and online. Thank you so much for joining us. Our next guest has arrived in the waiting room, so I must ask you to move on, but thank you so much. That was so interesting. And enjoy the rest of International Microorganism Day from Groningen. And yeah, thank you again. It's been great.
Thank you so much. See you guys around.
See you soon.